Salam sejahtera and a good day. Let's talk about genetics and nomenclature of rodents. Genetics and nomenclature of rodents are actually a very important part of rodent research because it enables us to understand the intricate nature of research using rodents. And at the same time, it also allows us to understand why certain model works and why certain models doesn't work. Let's talk about stocks and strains. According to the International Committee on Standardized Genetic Nomenclature for Mice, stocks refers to outbred animals, whereas strains refers to inbred animals. You can see there depicted on the screen a series of animals that are actually available online for ordering. These animals are actually made for a specific purpose. So in this case, most of them, for example, this 129 strain from Jackson's lab, it is actually an inbred strain. So as this AKR from Jackson's lab, how do we know that whether it's actually inbred or outbred? Just by looking at this / icon. We shall talk more about that in the subsequent slides. So why are strains and stocks important? They are important, like what I mentioned earlier, because it will determine whether certain animal models is actually suitable or works for the disease or the drugs that are being tested. Well, let's take a look into history. The DES sons and daughters refers to a series of fiasco associated with the diethylstilbestrol or DES, which is actually a synthetic estrogen. Believe it or not, it was actually approved for between 1940 to 1971 for treatments in women. At this age, at this day, I don't think we'll even think about giving estrogen to human beings because we know that there are plenty of receptors on certain types of tumors. They are actually promoted by estrogens. Well, back to the DES. It was aggressively marketed and promoted from 1953 to 1960s. And as you can see here, based on this ad from 1957, it actually promotes the use of DES in women to prevent abortion, miscarriage, and premature labor, something that is very, very concerning at that age and time. Well, why do they promote the use of DES at that stage? Because testing shows that, well, the drugs is safe. Wow, that is actually erroneous because at that point, they did not realize the differences between the strains and stocks, and different animals or different animal models will have different responses to drugs. Had the researchers had better access or a wider range of animals, they would not have concluded that DES is actually beneficial. So what would be the outcome of this kind of trial? Well, approximately between 5 to 10 million US women were exposed. And the drugs, in fact, was used in Europe until 1978. Why it's so dangerous? Because DES causes cancers, not in the first generation, but in the second generation. The consequence, the seriousness or the consequence of this application is clearly depicted in the CDC's website, depicted here, whereby they do track the DES mothers, daughters, sons, and until at this point, the third generation, further showcasing the ill effects or the poor judgment of using inappropriate strains and stocks back from the 1940s to 1950s to this day. As you can also see here that there are also law firms that offer the services to file a class action suits against the original manufacturer, which happens to be Abbott Laboratories. Abbott Laboratories is now no longer in business because they have been absorbed by another pharmaceutical company. So you can see here the far-reaching effects of using the wrong strains and stocks for drug testing. As a result, well, many women and their offsprings are actually suffering from cancers, and worse still, the cancers are actually found in the second generation. So those are data from the US and Europe. But what about Asia then? God knows how many women has been exposed to this type of drugs. So let's delve deeper into what causes all this. Based on the work by Shalabhadra et al. 1978, which entitled Red Differences in Memory Tumor Induction with Estrogen and Neutron Radiation, you can see here by using different strains of animals, different stocks of animals in this case, the incidence of memory tumor will be very different. So you can see here, if you are using an outbred SD rat stocks versus an inbred ACI rats, you will definitely see a very different, very stark difference in terms of incidence of memory tumor. So you can see here, when DS is given to the outbred Sprague-Dawley animals, the DS treated animals actually has much lesser incidence of memory tumors. But when it is given to ACI rats, which in certain respect is actually closer to engineered to be closer to human beings, you can see that DES actually promoted memory tumors up until almost 70 or 80%. So you can see here, if the researchers have access to these types of information, they wouldn't have allowed DES to be marketed. So the conclusion here, based on these findings, is that DES, well, safe to use if test was done as the rats. But then again, we have to ask ourselves, are the human physiology or the human genetic makeup is closer to the SD animals or the ACI animals? As you can see here, depicted in this gray print, in reality, DES administered to women resulted in increased cancer in their daughters and sons, with more than 250,000 of cancer cases recorded. That is only based on data from the US alone. We are not looking at data from other parts of the world. Okay, the second reason why they are actually important will be that, well, if you fail to take into account genetic difference between outbred animals or inbred animals when performing certain experiments, it could yield results that are not usable. For example, failure to take into account genetic difference between outbred Vista rats versus Vista Kyoto rats, which are actually in itself an inbred animals, when performing antibody labeling on microglials. Well, you may see from publication whereby they actually depicted or describe the type of animals that they use. There are reasons why inbred animals are being used, because inbred animals typically will have a much more uniform genetic background and therefore much more advantageous in antibody labeling. So this is actually just one of the reasons why you need to know strains and stocks. Secondly, there's a need to verify that the supplier has delivered the right stock or strain of animals. For example, long Evans rats depicted here in the lower left. These are animals with black or brown hood. And well, again, it is different from another long Evans or long Evans agouti or LEA, which is actually an inbred agouti, which has, agouti means that they actually has a triple color on the hair shafts also. Of course, on the outset, they appear very differently from their long Evans outbred cousins. And of course, the next important thing is that we need to know the best model for the work and the expected phenotypes. You may be wondering why certain types of animals die earlier. Certain strains and certain stocks are much more resilient, and so on and so forth. For example, you may want to start your work using the leukemic AKR mice. And if, let's say, you plan for a 2-year work, whereas in reality, the leukemic AKR mice strain has much more shorter lifespan than that, well, you could be in trouble. Because typically the leukemic AKR mice started dying at 6 months old, and at 1 year old, typically only about 10% of them survive, but even then is in poor condition. So knowing your strains and stocks are important because that will actually allow you to maximize the benefits of the animal models and to plan ahead on what you are going to do or what you're going to deal with later. So this is actually just a description for the AKR mice from JAX. As you can see here, depicted by this / mark, it means that these animals are actually inbred strains. So again, strains refer to inbred animals, stocks refers to outbred animals. As you can see here, there are also differences between strains and stocks in terms of a response to the disease model. As you can see here, in this case, this is actually a cast animal also from Jack's lab, but they're actually being used for flavivirus, okay, for flavivirus and so on and so forth. And flavivirus is actually important because there are a series of pathogens that are responsible for dengue, yellow fever, and of course, multiple forms of encephalitis. So if let's say we are trying to do dengue research and we are not aware about the advantages or disadvantages of certain mouse strains, we may not be able to replicate or we may not be able to grow the viruses in the animal itself. That's why it becomes very, very important for us to pay close attention to the strains and stocks being used in research, which is supposed to be clearly described in the methodology section of any reputable scientific paper. So these are further description. So I hope that everybody can just go on and read up about what is actually going on for the appropriateness, in terms of appropriateness of the strains and stocks for your respective work. Now, let's go deeper into what do we mean by outbred and inbred. As I mentioned earlier, outbred animals are also known as stocks. Stocks are typically preceded by the breeder code or somebody who holds that breed. And this is typically a 1 capital to lowercase descriptor. For example, here, H capital, S and D lower capital. And one key feature that distinguishes one particular type of animal as an outbred animal is the appearance of colon. Colon depict that this animal is actually an outbred animal. And beyond this, you do have another designation telling the brand name of the stock. And the brand name is typically followed by the origin stock name in brackets. For example, in this case, this animal is depicted as branded as CD of Sprague-Dawley origin, derived via foster nursing, F, and produced in a barrier type facility by Harlan Laboratories, USA. So Harlan Laboratories holds a breeder code of H capital, S, D in small letter. So there are many other breeder codes, for example, CRL, Charles River, BLU, Blue Spruce Farms, Teconics, and so many more. Producer's brand name for the stocks can be in many forms. It's just like when you have a car model, you can have a sedan car, but the sedan car comes in the brand of, let's say, a Japanese brand or European brand, American brand, Chinese brand, and so many other examples. But all of them are sedan cars. So in this case, you can see in the earlier example, the Sprague-Dawley is branded as a CD. And of course, other manufacturers, instead of calling their SD rats as CD, will have known to call them as N, CFE, SPD, ZM, and so on and so forth. So you can actually look up for more information on the respective manufacturer's website. Now, next to this name will be the origin of stocks. Several common ones will be Swiss mice, in capital S. Swiss Webster mice, SW, ICR, Institute of Cancer Research, and of course the ever famous SD or Sprague Dawley. Then the subsequent designation with derivation technique, either it could be fostered, egg transplant, ovary transplant, or just hand-bred. All these depict the degree of complexity in terms of the derivation and how how these animals originated. And after the derivation technique, of course, will be the hygienic state. This hygienic state is something that is very important because it will tell us whether these animals are actually a barrier house or SPF or notobiotics. You can see here on the lower right, in fluorescent, you have UPM, Ticus, SDBR. It just simply tells us that these animals, these Sprague Dawley rats, are branded as Ticus, and then they are being kept at UPM under a barrier facility. So from there, you can actually know a range of information about what the animals that you're using, what are the origins, and so on and so forth. In terms of inbred animals, it started with an inbred strain. Excuse me. And typically it's all capital or / or depicted in numbers. But the key feature is that it is depicted by a / If colon denotes outbred, / denote inbred animals. And the designation after the / are typically known as substrain. And substrain is typically given as per registered code except C in Bob C. Bob C is actually an inbred animal, as you can see here. The correct way to write Bob C animal is supposed to be B capital, A capital, A capital, B capital again, / C, whereby C denotes the allele. And the other famous one will be C57BL6, or typically known as a black 6 animal. These two are exceptions from the nomenclature standards prescribed for inbred animals because their names precede the inbred naming standards. And unique to the inbred animals, they do have a filial generation number indicating how many generations that has elapsed since the first time these animals have been bred. Now, let's look at this example below here. We know that this is actually an inbred animals because they actually a / mark here. And this is actually a Vista Kyoto inbred animals by Charles River, by the breeder code. As you can see, this is actually similar to the outbred stocks codes that you can see in the earlier slides. Bred from freeze-preserved embryos, P, and rats are germ-free, GF, and inbred for 80 generations. So F80 denotes 80 generations. This is something that is very important because an animal is only defined as inbred after 20 inbreeding generations. So there are further substrain designations in terms of the for the inbred animals. So you can see the substrain designation pertains to where these animals are being kept. It could be at the Harlan Lab, Charles River, Blue Spruce Farms, Jackson's Lab, denoted simply by J, Heston, and many more. And strain symbol are all in capital. For example, as you can see here, BALB or bag albino, or POPC, DBA, NZB, LE, and all kinds of other animals that follows. You can also have subline derivation techniques, which could be P for freeze preserve, F for fostered, E for egg transplant, O for ovary transplant, and H for hand reared. And of course, sometimes we may not know the inbreeding levels. Inbreeding levels are typically designated as F and the number of inbreeding generations. And this is typically stated as question mark if the number of generations are unknown. For example, question mark followed by 30 indicates that 30 generations since the last unknown generations animals were bred. And of course, sometimes instead of F, it may use N for back crossing. Backcrossing is being done when we want to derive a much better, when we want to derive certain features that that is similar to what we want to get from the parents. Now let's look at the stocks and strains of mice. Common outbred mouse stocks are Swiss, Swiss Webster, ICR, and of course common inbred mouse strains are Bob C. And this is actually something that I want everybody to pay attention to. ICR is not equivalent to Bob C. They may look the same, but ICR is supposed to be an outbred animal, whereas Bob C are supposed to be inbred animals. So this is actually important for us to know because it will allow us to have an accurate description of techniques and outcome that we supposed to be expected from these animals when you communicate, especially when you communicate with researchers outside Malaysia. And what are they typically being used for? You can see here, these mice are being used for a range of works on genetics all the way to infectious disease studies. There are over 400 documented inbred strains of mice, but all of them come from these original four, which are black albino, the C3H agouti, and then a C57 black 6 here, black, and the brown animal. The DBA animal is actually the oldest inbred strain, which was established way back in 1909, is actually a father of all this. In fact, one of the more recent strain of animal is actually the black albino. The black albino, believe it or not, the black albino was actually derived from a black animal here. And further description and strains of stocks of mice is actually being given on this slide itself. So you can see here, this is actually a stock because they are designated by colon. So you have CRL, CFW, SW. So these are CFW branded animals of Swiss Webster origin produced by Charles River Lab. And the descriptor here on their website tells us that it is actually bred by Dr. Leslie Webster, that's why Swiss Webster, at Rockefeller Institute in 1926. They are general purpose animals, but they are mainly being used for safety and efficacy testing. The next animal will be the CD1 branded ICR based animals, origin animals, bred at HSD, also an outbred stock. They are used for oncology, toxicology, vaccine, aging, and teratology research. This series of slides depict inbred animals, ranges from the black 6 from Jackson's lab, Heston, C3H, and they are, well, so-called most cancerous. Doesn't mean that they carry cancers, but they are very, very prone to cancer. They are developed from black albino crossed with diluted brown agouti, and typically 90% of them will have mammary tumor after 1 year. So this is really something, some feature that researchers prize because it allows them to do targeted cancer research. DBA, as what we mentioned earlier, is the oldest inbred mouse strain, and is a foundation strain for many others. And of course, the ever famous A strain, bred by Jacks, which was developed, which was it itself developed from black albino in 1921. Sometimes it becomes even more important for us to know that in order to have the animal used or depicted by the paper, you may need to have more than one strain of animals. For example, these animals, CD2F1 hybrid. CD2F1 hybrid is actually a very unique type of animals that are being used for safety and efficacy testing, transplantation research, monoclonal antibody production. And in order to have these type of animals, you need to have a BOC and DBA2. You cannot just simply have one, and only the F1 generation can be used. So that means you cannot keep these animals continuously and allow them to breed among themselves. So whenever you need these animals, you need to go back to the parent strain, and then after that, breed them and use the second generation or the filial 1 generation. Then again, the designation Y For example, BOP-C DBA2 means that the female line, the first strain should be the female line. And in this case, BOP-C should be the female line, whereas DBA2 should be the male. And you cannot flip this, the way that these animals are being bred. So this further underscores the importance of first to understand, okay, to understand the importance of using the right strains and the right types of animals. Let's move on to rats. The common outbred rat stocks are Sprague-Dawleys, Long Evans, and Wistar, WI. And common inbred strains are F344 or Fisher 344 for toxicology testing, especially and is being prescribed by the EPA, Environmental Protection Agency of the United States. So, and then we also have Lewis rats, Wistar Kyoto. Try to distinguish between Wistar Kyoto and Wistar Institute animals. Wistar Institute animals are actually outbred, whereas Wistar Kyoto are supposed to be inbred strain. As you can see here, these are common outbreeds. For example, the Sprague-Dawley branded Sprague-Dawley animals from HST. They are developed from single-hooded male and 6 albinos in 1925 in Sprague-Dawley Farms, Madison, Wisconsin. And that's why it becomes something that is actually very important for us to know what their origins and what they are being originally used for. Pay attention that the WI here indicates the American state rather than the Wistar Institute. And this is again the other one, the Wistar rats are actually foundation of all other stocks, and the name itself, WI, stands for Wistar Institute, Philadelphia, Pennsylvania. More information about the strains in rats. So F344 is actually a general purpose strain for cancer research, and then the females are susceptible to mammary tumors, while males to testicular institutional cell tumors. They are also being used for a lot of toxicology testing. Lewis animals are developed by Dr. Margaret Lewis from Wistar Institute in 1954. They are susceptible to inflammatory diseases, glomerular damages, unlike the resilient Sprague-Dawley animals. They are being used for lymphomas, mammary tumors, and also in skin disease and skin grafting research. They have much more shorter lifespan due to neoplasms, which typically has an average lifespan of about 26 months. SCI animals are a very good model for liver cancer and other liver research, and they have known renal problems at old age. They typically have very poor reproductive performance, high congenital deformities, and high in utero deaths. These animals were developed in 1926 by crossing a Sagalov August rat to a Copenhagen 2331 female at Columbia University. Of course, the ever-famous WKY, developed from the Kyoto School of Medicine, imported to the National Institute of Health in 1971, and ever since that point, it has been famously used for hypertension and cardiac abnormalities research. This is actually unique because this is actually an inbred animal developed from an outbred animal. So having known these stocks and strains of mice and rats, so what's next? Well, we have to know that the future description of mice and strains requires a good understanding of genetics. So before we move on, we also need to know that there are certain other descriptors of the stocks and strains. You could encounter congening animals. Congening animals are animals that differ from another animal within a strain by one particular locus or a limited region within genome. And of course, sometimes they are also known as consomic if it's differed by chromosome. So you can see congenic are animals differ from other animals within strain by one particular locus, whereas consomic if the animals differed by one chromosome. So these are important features that we need to know in order to allow us to study how genetic background affect a gene in an organism, as you can see here depicted within all this diagram here. And you can also have another type of animals known as co-isogenic animals. These are animals that differ from another animals within strain by one mutant gene due to spontaneous mutation. This is actually important to study how a gene mutation affects organism. So contrast this with an earlier slide whereby we want to study how genetic background affect a gene in an organism. So this one, the core isogenic animals, is actually important for us to study how a gene mutation affects organism. So this series of slides serves only as introductory primer to whatever that you may encounter later in nomenclature. First things first, always remember that outbred is indicated by a colon, inbred indicated by a /. And from that point onwards, you can have many other names. For example, congenic animals, like what I mentioned just now, is indicated by a semicolon. And then again, you can have a semicolon for animals that have been back crossed for less than 5 generations. More than 5 generations, instead of colon, is denoted by a dot. So you can see it becomes something that is rather complicated here, and something that we really need to pay attention to for us to know. I'm very sure a lot of us have encountered the more than 5 generation congenic animals in publications, because these are the common animals that we commonly see in research. And of course, you may see also certain other animals used, for example, in this case, used for lipid research that contains a very specific locus of interest. For example, apolipoprotein A1 gene in the black 6 animals bred to 129 animals background, as in this case. You can also have targeted mutation and denoted by TM, in this case, targeted mutation for apolipoprotein A1 gene in black 6 and 129 animals. This one tells you there are 2 strains of animals here, C57BL6 and the 129 mice. Okay, so and this, in this case, it was developed at the University of North Carolina, but being kept at Jackson's lab because /J here. And in this case, TM1 tells you that it is actually a foundation line number 1. You can also have transgenic animals whereby a foreign gene is actually being inserted. In this case, is indicated by TG. So the TG here, so for example, C57BL6, TG, CAG, EGFP1, OSB /J. It tells you that this is actually a transgene. Okay, this transgene CAG from human cytomegalovirus and chicken, rabbit, a jellyfish were incorporated in Foundation 1 by the short form of Dr. Masaru Okabe, developed at Osaka University. But eventually, these animals are being kept at Jackson's lab. So /J, because as you remember, / indicates the subline and derivation. So you can see these are all rather complex naming schemes. So I do hope that with the information that I already provided to you today, you shall be able to start to look at, maybe understand a more complex naming schemes, which in turn is actually very, very important for us to understand the proper animal models used for animal research. I think with that, I thank you for your attention, and I'll see you in the next session.